Okay, so I think it will be Thomas. So Thomas Hillen has been very, very pivotal to helping us get this workshop together. So thank you for that. And he's going to be talking to us about, well, oncolytic virotherapy.
Yeah, thank you very much. So don't worry, I will not use a double time, although maybe I could. And to be clear, this is not a project, so nobody is allowed to work on this, okay? It's just but, well, we do have a project, you're right, exactly. So it has directly to do with this, yeah. So this is joint work with Awab Abdullah. She finished PhD and now she is professor at United Arab Emirates University. And she's also online, I think. I saw her there. Yeah, there she is. Negar Mohammad Nejad is a beginning PhD student. She is right here somewhere there. Wonderful. And then Morgan van Balsam, she was an undergrad student working in my lab over the summer. And yeah, so in the first half of my 10-minute talk, I talk a little bit about the standard oncolytic virus model. So there is a standard model. And many people in the room have worked on this. Peter Kim has written a nice paper recently, and Tracy Jackson has written a few papers on this. And then in the second half of my 10-minute talk, I talk about an application to a reovirus and how to make reovirus more efficient for cancer treatment. Okay, so that's the idea. We have seen a similar picture before in Dr. Yoon's talk. So there are essentially two effects of virotherapy of cancer. The one is a direct kill. So the virus infects cancer cells, and then the virus reproduces inside the cell, and then the cell lyses and releases new virus particles. But the other effect is the marking. So the virus just marks the cancer cells as infected, and then the immune system comes in and kills everything which has the marker of the virus. And this is good on the one hand, but on the other hand, it's a bottleneck. Because if the immune system comes in too quickly, then the virus did maybe not have a time to infect all the cancer cells or find all the cancer cells. And then the immune system would actually stop the effect of this oncolytic virotherapy. So I think the key is you need to find a virus which is fast enough. It spreads enough and is potent enough, and the burst size has to be large enough so it can infect all the cancer cells before the immune response kicked in. Okay. So as I said, there's lots of work being done on oncolytic viral therapy modeling, and here are important names in this area. And this all comes down to a relatively standard model, which is this. So here we have equations for the cancer cells, infected cells, and virus particles. And in this case, I use standard diffusion for the spatial spread of all these. We have a logistic growth and a competition term here between the cancer cells and the infected cells. There's an infection term. And then this here is important. B is the burst size. So this is how new virus particles arise. So first thing you can do is a non-dimensionalization, get rid of a whole bunch of parameters. Now, if you look at this a little bit more, then you will notice that this is a predator-prey model. And predator-prey models have been analyzed a lot in the literature and also in textbooks. So predator-prey models, what do? Well, they have oscillations, right? They have oscillations, and this is what this model has as well. Oh yeah, so I look at the ODE part first, no diffusion, and then this is a very typical bifurcation diagram for these type of models. I show two curves because we compare a model by Tian and our base model. So the red curve is the model I'm showing. The Tian model has a little variation, which I'm not bothered about today. But anyway, so here's the burst size B. And as I increase the burst size, first of all, I have the cancer state is stable, but then you get a transcritical bifurcation and you get a coexistence steady state. And then here is a Hopf bifurcation, and then you get these oscillations happening. So as function of time, they look like this. So on the left, we have, what do we have? Blue is virus goes to 0, cancer survives. In the middle, we have an oscillating approach. We have a stable spiral. And on the right, we have the periodic orbits. And so what we realized here is that the cancer density goes very, very close to 0. And then the question is, can we consider this to be eradicated, or is it an infinite period? And so we looked at the tumor control probability. So the tumor control probability is the idea, how likely is it that the tumor is actually eradicated if you have a very small number of tumor cells. And you see here on the left, yeah, the TCP rises first to 1, but then it settles somewhere in the range of 40%. Whereas here between the oscillations, we get a plateau at the tumor control probability 1. So that leads us to say that the periodic case is actually the good case, because between the periods, you come very close to 0, and the likelihood to cure the cancer is actually very high. Okay. Yeah, now let's include space. And I think I want to do this. Yeah. And we do this in 2 dimensions and just see what this model does in 2D. So then we initiate the virus in the middle here. And what you see is a ring spreading out over the cancer cells. So the blue background is all the cancer cells. And it runs out of the domain and then starts again. And then runs out of the domain and starts again. So you see a beautiful period also in the spatial context. But that's not all. So if you run it very long, then all hell breaks loose. And you get very irregular patterns. And we thought, or we first thought, maybe our numeric sucks and we have to use a better numerical code, which we then did. And it comes back to the question, what happens to coupled oscillators in a spatial context, right? And in 2D, at every location, you have an oscillator, and now they interact and they oscillate, and so you're nodding your head, so you know what's happening, I guess. You can get all kind of chaotic patterns. So here's an example where we start with 5 inoculations here in the middle. We get this concentric kind of rings, but then they deteriorate into very, very complex patterns. So this is mathematically really interesting, I thought. Biologically, though, I mean, T is 1,000. This corresponds to about 1,000 days. And the immune response kicks in about 5 days. So I think this is not very relevant for the applications, but the short term is probably more relevant. Anyway, so then comes Maya Shmulevitz. She's at the Li Ka-shing Institute of Virology at University of Alberta, and she designs reoviruses for oncolytic virotherapy. So she takes reoviruses, she does mutations, increasing the burst size, increasing the binding rate, et cetera, and designs the efficiency of these viruses. And she had an interesting question. So the belief was, if you increase the binding rate of the virus, then this is good for the therapy because the virus can bind to more cancer cells and you get a better effect. And she had 2 viruses, the wild type and the mutated one. And the mutated one had a higher binding rate and it was more efficient on cancer cells. And now she wanted to know, is this the best I can do? Is there an optimum or can I further increase the binding rate, etc.? And then when I visited her in her office, it's a virology office, right? So I thought everybody has is fully covered in white aprons and has masks and gloves and whatever and high security and you need a thumbprint to get in. But no, I was sitting at her desk and she showed me exactly this petri dish. And she said, yeah, this is a virus. And it was an open petri dish. And she just showed it to me. They're like, what? So she said, it's totally harmless for people. And so, yeah, I had to believe her. Okay. Anyway, so I saw these pictures and I thought immediately, well, we have this fancy oncolytic virus model. We can do all these patterns. So doing these little dots is a piece of cake. Can do this in an afternoon. Yeah, of course, it didn't work. So we tried a lot and we tried to model, to use our model for these data. And we were not very successful. So we used quite some time, but it didn't work. And it didn't work because the data didn't fit to our model. I mean, how could they? How could they measure data which do not fit to our model? Ridiculous. So, but then I remembered something which Heiko said to me. I don't know if Heiko is here or just left. Anyways, so he said that simple models are sometimes better than complicated models. So if you have a set of data and a simple model can explain the data, then why not? Just use the simple models. And so then we did this approach. And so, for example, for the binding rate, so they had experiments where they measure the binding rate. We just used a linear model for exponential decay. And they had experiments for viral spread on agar. So we used just a simple diffusion model for this. And then estimating the spreading speed of the virus inside the experiments, we used a model for infected cells and virus cells. So these are all kind of simplifications or little bits from the bigger model, which I have shown earlier. And then for the plug size, we could then finally use this model, which is, again, a simplification of the model I've shown earlier. And it worked just well. And then we not only fit the data, we then had other experiments where we could validate that the model does a good job. Okay. And so then the main outcome is this here. So we could then use it to answer Maya's question of if there is an optimum for the binding rate or not. So let's look at the which one? At the purple, at the red curve first. Is this pointer working? Yeah. So the red curve here. Here we have the binding rate, and there we have C star, which is the invasion speed. So how efficiently the virus spreads over the cancer cells. And there's a clear optimum. So if the binding rate is too low, we get almost no spread. If the binding rate is too high, it goes down as well. And the optimum value here is the black dot. And the red value indicated here is the one which they measured in the experiments. And they had a second virus type, which is the purple one, SV5, which is up here. And then again, their measured binding rate is here, and the optimum is here. So we could show them, first of all, that there is an optimum. And secondly, that with the SV5, they were actually very, very close. I mean, they might improve just a little bit, but for the spread rate, there's not much gain, but at least a little bit of a gain. Okay. Yeah, so this work is submitted right now at the journal and also on the bioRxiv. And then that's the end of my talk. Thank you very much.
Thank you. Questions for Thomas? Okay, well, I'm going to ask a question.